The next learning outcome in microbial control, physical methods of control, is explain how high osmotic pressure, desiccation, and freeze drying are used to control microbial growth. So if we just look at those terms, high osmotic pressure, that's going to be removing water, right? High osmotic pressure, hypertonic environments, things like adding salt or sugar or just drying things out would put cells into a situation where they have high osmotic pressure. Desiccation simply means removing water. So here's an example of high osmotic pressure. They added salt to fish, or you added salt to ham, salt to meat to make ham. Desiccation would be just drying apricots, leaving them out in the sun or in a dryer and letting the water be pulled out. Or freeze drying is a special technique where the in this case strawberries are flash frozen, and then there's a technique that pulls the water out of it. So with freeze dried things, they're kind of crispy and they retain their shape. With something that's just desiccated, it's going to be kind of chewy and moist. But freeze drying is going to be kind of crispy. And but all of these leave the food in a situation where they have high osmotic pressure. They have high solute and very low water. And you can imagine that having very low water, you cannot conduct metabolism. So, so the reason why we've made things like dried fish and dried fruit and hams and bacons and things like that is they've all been developed as ways to preserve food. They allow the food to last longer because you discourage the growth of microorganisms. So it says high osmotic pressure, desiccation, and freeze drying can all be bactericidal to some degree, but they're mostly bacteriostatic. So they don't sterilize. They can kill some microorganisms, but there's probably some microorganisms still there, and they might even be alive. They're just not growing. They're just kind of hanging out there. Okay, so these situations, the foods are bacteriostatic. I like to point that out because if you ever get like freeze-dried food when you go camping, some people will buy those freeze-dried meals, and as soon as you rehydrate it, it's no longer preserved. It's the microbes that were there were not growing. Well, once you add water, the microbes get rehydrated and they can grow, and that food can spoil just like any other food. So these foods, there can be microbes associated with them, but they're just not growing. It's bacteriostatic. Let's see, where are we? In fact, freeze drying can be used to store bacterial cultures. Sometimes I'll order a bacterial culture from a company and it comes as a little dried out disc. If you freeze things very quickly, freeze dry them very quickly, you can actually, it's much more bacteriostatic than bactericidal. So it's actually a way they use to preserve bacteria. But in the case of food, it doesn't matter if there's some bacteria there. We just don't want a lot of it, and we don't want the wrong kinds. So freeze-dried is just fine. Another physical control method has to do with radiation. And it says differentiate between ionizing and non-ionizing radiation. It says microwaves, but I meant to take that out. We have that later on. Oh, no, we do it. No, I'm sorry. I moved it to here. Differentiate between ionizing and non-ionizing radiation and microwaves, and how each can be used for microbial control. So I put this here just to give you, you've probably seen this somewhere before, but a visual of the light spectrum. So light is in these waves. We have waves of light, and visible light goes from 400 to 750 nanometers. So waves that come in the visible range, radiation that comes in this range of 400 to 750 nanometers is what we can see. So there's all the colors of the light. But there's also wavelengths that we can't see. And so that gets beyond here. Ultraviolet light, X-rays, gamma rays. You've heard of infrared light, microwaves, radio waves. And so you can see this little range is our visible light. As we go into lower wavelengths, these wavelengths have higher energy. And some of those can be used for microbial control. So if you look down here, first of all, we have the UV light in the range of the sun. And here's the range at which you would be getting tanned. And down here, that where the UV or ultraviolet light is bactericidal. So we call this ultraviolet light, UV light, these wavelengths that are right below visible light, as non-ionizing radiation, also called ultraviolet light. So wavelengths in this range, you don't have to remember what range, but just remember that non-ionizing radiation slash UV light, they're the same thing, can be used for microbial control. It damages DNA. If you look here, remember sunlight can cause cancer. Why does sunlight cause cancer? Because it damages the DNA in our skin. But it doesn't cause liver cancer. It doesn't cause lung cancer, because UV light, having this relatively low energy, doesn't penetrate. Just like visible light. When you shine a light on your hand, you illuminate the surface of your skin, but you don't illuminate the inside. The wavelengths of light just aren't strong enough to get through your skin. The same thing with UV light, it just gets to the surface. So if you want to use ultraviolet light to kill bacteria, and you can use it to sterilize surfaces, if you want to use UV light, it's only going to sterilize a surface. And I'll show you an example of that, because it can't penetrate. Somehow, oh, I skipped ionizing. Here we go. Ionizing radiation is more intense. So here we said we go this way and we get stronger energy. So X-rays and gamma rays are what we call ionizing radiation. And these wavelengths are more penetrating. They're much more potent, and they also damage DNA, but they can go through things. So if we go, the only way you get exposed to X-rays and gamma rays are, we can generate them through equipment, and we can make X-rays, as you know, and make gamma rays, and they will penetrate right through our skin. And that's why we can see our bones when we do X-rays. And so they also can damage DNA, and they can also be used for microbial control. And both ionizing and non-ionizing radiation can cause cancer. So these are, and can sterilize. I'm sorry. They can both cause cancer, but they can both sterilize, because they can kill endospores. Because endospores have some DNA, and they can penetrate the outside of the endospore and damage endospores. So these can be used to sterilize. I'm going to show you some. Oh, the last thing I want to mention on this page is microwaves. And microwaves are up here. And they, as you know, we use microwaves to heat up our food. And there was some talk years ago that got around that people said, oh, you can use your microwave to sterilize your sponge. And so people started putting their sponges in the microwave, and what happened is their sponges caught on fire, and people ruined their microwaves and had all sorts of problems. And it turns out microwaves themselves are not antimicrobial. They don't damage bacteria. What they do is they heat things up. So you put your wet sponge in the microwave and it heats it up, and that hot water that's generated can kill microbes in your sponge. But once that hot water evaporates, now you're just heating the sponge, and the sponge can catch on fire. So it is not recommended that you use the microwave for microbial control. You can use heat for microbial control, and heating something up in the microwave, you heat up soup and it's hot enough and you're going to kill some microbes, but microwave is not a reliable way to kill microbes. I wanted to give you some examples of how we use gamma rays for microbial control. And you've seen in the virtual labs, and we do this in the live labs, that we use these petri dishes. They're plastic. And remember, I said if you put one of these in the autoclave, it will melt. So I buy them. You buy them in these plastic sleeves. They've already been sterilized, and you've got like 24 plastic sleeves inside of a big box. And so what they do is they take those big boxes and they stick them in a big chamber where they can generate gamma rays. And obviously it would be only things inside of the chamber would be getting exposed to the gamma rays, because you could kill people very quickly with gamma rays. They're that dangerous. But you can expose things like petri dishes to gamma rays. And then all of a sudden you say, oh, yuck, I don't want to touch petri dishes that have been exposed to gamma rays. But remember, it's just like exposing it to light. You turn on the light, and then it's illuminated. Once you turn it off, it goes away. You don't have residual radiation on this. Pipettes that we use in the laboratory, they're all gamma irradiated. They're already put in these plastic bags, and then they're sterilized. Wegmans has in the freezer section irradiated ground beef. And it's just a way that some people want to buy ground beef to let it last longer. I was reading an article that said if you take regular ground beef, that you buy in the meat section, and you leave it in your refrigerator, in about 5 days, it's going to be the microbes are going to grow enough that it will not be good to eat. Once you open this irradiated ground beef, it still is shelf stable for about 42 more days. So the only microbes that are going to get in there are the ones that will get in there once you've opened it. So this is a way, using irradiated ground beef is a way to extend the shelf life. Um, Again, people say, oh, I don't want to eat food that's been irradiated. But the thing is, there's no radiation residual in the food. It's just it's completely gone. The danger with it is that, oh, if I'm going to irradiate a lot of food, I need a lot of radiation plants where we have these gamma rays, and gamma rays can, they're dangerous. They're not leaking out of the plant, but you could see somebody could have an accident and release radiation in a plant. Anyway, not likely to happen, but it could. Where we use UV light a lot, working in research labs, every microbiology lab I've ever worked in has a chamber like this. It's called a laminar flow hood. You don't need to know that. But what this is, it's a place where you can, it blows sterile air over your whatever you're working on. So the air in here is sterile. It's free of microbes. So you see she's got this open, and she's not worried about bacteria getting in it because the air inside here is sterile. And then what you do is, when you're all done, you pick everything up and clean it up, and then you turn on an ultraviolet light. And that will sterilize anything on the surfaces that got in here. So in research labs, a lot of times we'll use ultraviolet light to sterilize a surface. Okay, remember, ultraviolet light only affects surfaces. You don't want to be in there. You don't want your hands in there. You'll get a really bad sunburn really fast. But you can use an ultraviolet light to sterilize surfaces. Another place, it's getting relatively common for things like apple cider to be pasteurized using UV radiation, UV light. And instead of running it through, instead of heating it up, they run it through a specially designed tool that will reduce the microbes in the apple cider. And some people say the apple cider doesn't taste as good if you heat it up. So it can preserve the food quality if you just expose it to light. Some people have chronic bacteria problems in their water at home. And so they might have they might UV irradiate their water. And so here's how you would do like apple cider or water, that you run the water through this device, and there's a UV light in the middle of it, and the water runs in a thin stream around that light, and the microbes are killed. So there are ways that ultraviolet light can be used to effectively kill microorganisms. This is one, and I put 2018 to remind myself. I had a student that was working at Corning Hospital, and he said they were using this Xenex robot. And what it does is, after a patient's been in a room, let's say you have a patient that had tuberculosis, or they had COVID, or they had, any infectious disease. They might have had, I'm thinking of, just left my head. But anyway, there's all sorts of infectious disease. C. difficile, I'm thinking of the fecal bacteria that can contaminate. It's a real contagious and really um, common fecal digestive system infection, C. diff. But it's caused by nasty bacteria that makes endospores. And so what they do is, in these rooms, they send in the Xenex robot. In front of the room, they put these signs like "Do not enter," "Do not go in there," because what this does is, once the robot gets in there, you seal it off so no people go in there. It lifts up, and then it shines a UV light through the room, and it moves around, and it sterilizes the surfaces. So if you had a patient in there with C. diff, which Clostridium causes, makes endospores, the UV light can kill any endospores that are on a surface. It's not going to kill endospores that are under something or behind something, but it's going to surface decontaminate. And these are being used much more frequently, primarily because it helps us reduce the number of antibiotic resistant bacteria in an environment. So let's see. I think I'm going to stop right there, and we'll finish up in one more video.